In this video, we are going to be looking at cancer cells and how they divide uncontrollably. In doing so, we'll describe how chemical signals regulate cell division. We will describe internal checkpoints in the cell cycle. We will describe what happens with cell division and when cell division and cell death are improperly regulated. We'll describe forms of cancer. We'll compare and contrast cancer cells and normal cells. We will describe how cancer treatments remove or kill abnormal cells. And we will describe how lifestyle, family history, and age influence cancer risk. So first, we know that cancer cells are abnormal cells. So compared to regular body cells, they do have some features that kind of stand out. First of all, they tend to lose their specialization. So they may structurally look different than the normal cells from which they originated. And that allows them to be picked out under a microscope, basically, if someone is screening visually for cancers. They are immortal. They can continue to divide endlessly. So that's one of the things that makes cancers, if you think about having a tumor or cancer spreading through a body, that's because cell division is going unchecked and the cell just keeps dividing and dividing and dividing, and there's no stop to it. So it never gets to that G1 phase or an S phase or anything like that. It just keeps dividing. They can regenerate the ends of their chromosomes. So the ends of chromosomes contain structures called telomeres. And over time in normal cells, as each time the cell divides, little bits of the telomeres are broken off of the ends of the chromosomes. This doesn't typically happen in a cancer cell. They will continue to recopy the entire chromosome, and the entire structure of that chromosome is maintained, so the telomeres are there. There's an enzyme called telomerase that is not functioning in cancer cells. Cancer cells also send signals for that stimulate a process called angiogenesis. This means that cancer cells can actually grow their own blood supply, which nourishes them and allows them to keep up that rampant cell division. And one thing that cancer cells have also that allows them to form things like tumors is a cancer cell, unlike a normal cell, does not have contact inhibition. So contact inhibition means that a normal cell, when it's in contact with another cell, that tells it that it doesn't need to grow and divide. And so a cancer cell will continue to grow, and they'll continue to grow and continue to grow. And that's why you might get a tumor-like mass, because they're absolutely not inhibited in their growth and development by contact with other cells. So the cell cycle is typically kept under tight control, and there are a series of chemical checkpoints that regulate this cell cycle. The checkpoints ensure that all the DNA has replicated, the DNA is not damaged, and that the chromosomes line up and separate properly. So let's start down at the G0 phase. Remember that that's the phase that most of your cells are in, sort of a resting phase. They go in and out of G1 and G0 depending on what the cell needs. So if the cell is having to perform a function or make some proteins, or it's growing, that's going to all happen in the G1 phase. At the end of G1, there's a checkpoint that stops and checks to make sure that DNA is the DNA damaged. If DNA is damaged, then there is a p53 protein that can actually encourage the cell to undergo apoptosis, apoptosis. Apoptosis is when cells are programmed to die, so it's programmed cell death. In the event everything is fine at the G1 checkpoint, or so the cell thinks, it moves into the S phase, which is DNA replication. And at the end of S phase, before the cell goes into the G2 phase, there's an S phase checkpoint that checks to make sure that the DNA replicated correctly. Again, if there are errors, the p53 gene can come in and trigger apoptosis. G2 is that additional growth, and this is when the cell is preparing to divide. So at the G2 checkpoint, the cell is making sure that all the DNA has been replicated. If there is any damaged DNA, can it be repaired? And is the spindle making machinery in place? Because remember, that spindle is what's going to be responsible for helping the chromosomes not only align properly along the metaphase plate, but also to be able to separate so that you get the proper DNA in each of the new daughter cells. There is a metaphase checkpoint, so actually during cell division itself, remember we had prophase, prometaphase, and metaphase. Again, this is when all the chromosomes are lined up along the equator, the central portion of the cell. Do kinetochores attached to the spindle? So are all of those chromosomes attached to a spindle? Are the chromosomes aligned down the equator? Remember, when the cell divides, we want half of the genetic information to go to each cell. So that's sort of your final checkpoint. So all these checkpoints sort of are stopping points to make sure everything is going well with the cell. If things are not going well, then the p53 protein will come in and trigger apoptosis or programmed cell death. All these checkpoints can go wrong. So one can go wrong, multiple ones can go wrong. Um, but ultimately, for cancer cells, it allows them to divide out of control. So if the body loses control of cell division, then the cells just continue to divide and divide and divide. And even if they are not needed, they continue to divide, but they also override the checkpoints. So when we're looking at a tumor that has potentially formed, so cancer cells, they divide out of control, and that mass that they form of a whole bunch of mutated or broken cells is called a tumor. Tumors can either be benign or they can be malignant. So in a benign tumor, the reason it's benign or non-spreading is because there's a capsule of connective tissue that surrounds the cancerous cells and keeps them contained. Now, it can continue to expand, but it's not going to spread to other parts of the body. A malignant tumor can undergo metastasis, which means that it can break off little cell fragments or seed new tumors throughout the body. So this would be how cancer cells spread. So when you hear about people who have different phases of cancer, one of the things that we know is that for example, if you have maybe you have a tumor in, let's say, in your liver, and pieces of that tumor break off and they can spread to the lungs and the brain, etc. So that's called metastasis, and that is tumor cells spreading throughout the body, and that is again a malignant tumor when it spreads. So the reason that cancer happens is because genes are mutated. So you can imagine cell replication is going wrong, cell division goes wrong, all those different checkpoints that we had, something can happen that causes the DNA to be mutated. So they alter the key cell control genes. And some of the things that may happen is that cancer can come from an overactive proto-oncogene. So a normal proto-oncogene, it is going to have proteins that stimulate cell division and results in normal cells. An oncogene, as compared to a proto-oncogene, an oncogene actually is mutated or overly active, and so it accelerates the cell cycle. Again, we get a whole bunch of division. We also have a normal cell has what's called a tumor suppressor gene. So a normal cell, the proteins of the tumor suppressor gene that are coded, the proteins that are coded by that tumor suppressor gene, they block cancer development. So they tell a cell that it's going to follow these various different checkpoints throughout its cell cycle. Tumor suppressor genes, however, can be silenced, in which case mutations occur that the proteins are not being produced, so they are not then blocking cancer development, and again, you get a mass of cancer cells. Cancer risk can vary by ethnicity because gene mutations can be inherited. Not all are. Some gene mutations happen in somatic cells, and so they are not spread. But remember, the ones that happen in gametes or sex cells, they are spread to future offspring. So a gene that could be encoded might be something like BRCA1, which is the breast cancer gene. And you notice that in this graph, you see that it increases, you see an increased risk of having the BRCA gene among the Ashkenazi Jews. I hope I said that correctly. Basically, they lived in small cultural groups and didn't tend to marry outside the group. So you have what's called the founder effect, which means that because that gene was found in some of the original members of that population, because there was a lot of intermarrying and because they were isolated sort of as a culture, that gene just got passed around throughout that group, and so they have a much higher risk of breast cancer. So some of the damage ways that you can avoid cancers would be to avoid DNA damage. Obviously, we have some inherited factors that we cannot avoid, but some factors in the environment can increase cancer risks. Some of the things that we can do to avoid or reduce the risk of cancer would be to eat a healthy diet, maintain a healthy body weight, and get a lot of exercise. Don't use tobacco products. Avoid UV radiation because ultraviolet radiation directly damages the DNA in our skin cells. Use self-tests and medical exams to detect cancers early. So a lot of people go for regular screenings, whether it's for skin cancer or cervical cancer. And then use contraceptive protection such as condoms, to avoid exposure to viruses such as the human papillomavirus, which can cause cervical cancer. So there are several different types of cancer treatments, and removing tumors is not enough. So before I talk about cancer treatments, let's just run through where um, like how severe a cancer is. So we have stage 1 through stage 4 cancer. In a stage 1 cancer, the cancer is pretty much just isolated. So it may be a tumor at this point in time. And in stage 1, the cancer has not spread um, to adjacent tissues. In stage 2, it has spread to tissues, but only in a sort of a limited area. When a cancer gets to stage 3, it starts to spread to organs and to lymph nodes, which then allows it to spread further. And then in stage 4, the cancers have spread to distant sites from the original location. So this would be, again, an example of a liver cancer releasing malignant cells that then could travel to the brain or to the lungs or to the bones or anywhere else in the body. And there are other cancers that can do that as well. The lower the stage, the better the prognosis or the better outcome for the patient. So, if someone is detected with cancer, what are their doctors going to do? Well, the first thing is surgery. So, surgery can remove tumors, but some cells with DNA mutations may have already spread. So, in that case, a broader ranging treatment has to be used, such as chemotherapy and radiation. So, chemotherapy uses chemicals typically to target specific types of cells, and then radiation uses intense radioactive energy to target specific locations of cells. Um, Chemotherapy, because it is attacking, sort of not attacking, but it's treating cells kind of in the cell division phase. This would be why things happen like hair falling out as a common side effect, because hair follicles have a lot of cell division going on. Your hair is constantly growing. So anywhere that there's constant growing, you're going to see some effect from chemotherapy. And then obviously radiation has its own cancer risks itself, but it is a good way to target cells. Frequently, and to everyone's benefit, immunotherapy and gene therapy have a lot of new types of methods that are much more selective, much less invasive, and damaging to the patient. So, immunotherapy is looking at targeting a cancer specific molecule. So, we've seen a lot of advancements specifically with leukemia, where there are immune promoting therapies that go out and specifically recognize and attack leukemia cancer cells. Others, in terms of gene therapy, there's a lot of hope for this for the future, where we could take a genetic code from a healthy cell and insert it into a cancer cell. So then, as the cancer cell is dividing, it will pass on the corrected copy of the genetic code. So to wrap up, what's wrong with cancer cells, and then how is cancer both a genetic and environmentally influenced disease? So cancer cells, remember, they basically are violating all of the checkpoints. So their cell division is going unchecked. In terms of being genetic and environmentally influenced, there is a genetic component to cancers. They can be passed down if they are in the gametes. So if they are something like the BRCA gene, which can be inherited, um, they also may put people at risk of being more environmentally influenced. So you may have higher chances of developing a cancer, but if you make poor life choices, then you can greatly increase the risk of coming down with that cancer. Just because you have a gene doesn't necessarily mean you'll get that cancer, but it does mean that there's a higher likelihood. Environmentally influenced, if you put yourself at risk by participating in risk factors such as lack of exercise, obesity, smoking. Those are all things that environmentally influence cancers. So, in this lesson, we talked about how chemical signals regulate cell division. We talked about the checkpoints in the cell cycle. We described what happens when cells divide and when cell death is improperly regulated. We described different forms of cancer and the different stages. And whether it was a benign or a malignant. We compared and contrasted cancer cells and normal cells. We described how cancer treatments remove or kill abnormal cells, and we described some of the risk factors of cancer.